Alright, so this video is going to focus on bacterial growth and bacterial cell division. So growth is kind of a strange thing to think about in bacteria. Alright, not that they don't grow, but really hard to see and measure, right? So you start with something that is, let's say that it's, 2 microns. So 2 micrometers, and let's say that it grows to be 4 micrometers, and it's going to look, let's even just say it looks more pill-shaped, right? So more of a rod than this coccus here. So let's just say that that's our growth. So this is just a generic example. Well, we can't really see that kind of growth, right? It's not like when you were a little kid, right? And you were and your parents, they had a wall at home, right? And they said, oh, look, you're this tall now. You are 4 feet, 3 inches tall. Hooray, you're getting bigger. Well, you just can't do that with the bacteria, right? They don't work that way because we can't see them. And it happens so fast, right? You've been doing growth curves in lab, and you'll see that, your reproductive time really could be anywhere between 20 minutes and an hour. And so, you got these tiny things, and you just can't see it. So what we're going to really look at then in terms of growth, and we'll talk about some of the things that happen in terms of enlarging size. So, so you can talk about growth, right? And you can say, okay, it's an enlargement of the cell, right? You're going to produce cytoplasm, right? You're going to replicate the DNA. Oh, but now, if we say that, now we're actually switching from growth to reproduction, right? And so really, when we talk growth, we usually talk about a population, all right? So when we talk about bacterial growth, for the most part, we're talking about a population, and we're talking about their ability to increase in number, okay? That's really what we're talking about. So if we look here, we've got a young cell at early phase of a life cycle, and so you can see here, right, it is going to grow. It is going to get bigger, all right, from there to there, right? It's just going to grow in size. Now, what's going to happen is that when this cell reaches a certain size, and so we'll notice here it must have reached a specific length, all right, in this second spot here, and that will trigger the reproduction of the chromosome. And so our chromosome 1 is in red and our chromosome 2 is in blue. And we're going to produce plenty of ribosomes. We've got to continually make cytoplasm, all right? And then we have to partition these 2 chromosomes when they are completed, all right? And so we're going to have to push or pull, right, these chromosomes apart. And we need a septum to grow in between, all right? And so what will happen is a septum is synthesized, and it is going to create 2 cell chambers, and then they will actually physically have to be divided here. And we get what we call 2 daughter cells. Now, this is very different than when we think about reproduction in humans or animals and things, in that this is not parent and offspring. This cell and this cell are the same age, in that in theory half of the DNA is old, half the DNA is new. They should have each received ribosomes, of certain, a group of them that are basically the same average age. And so really these two, if they are dividing, they are the same age, which is kind of an interesting thing to think about. So bacterial cells then, in reality, if it sat here for days, months, you could have an old cell, right? In terms of age. But if it's replicating every 20 minutes, the oldest cell in your culture is 20 minutes old, all right? Which is just kind of a fun thing to think about. So we call this process, binary fission. And binary fission just refers to making clones or copies, all right? And so these 2 cells at the bottom, right, these two here, they are, in theory, they are copies or clones of each other. Okay, and so that's binary fission. Now here, we want to focus a little bit more on E. coli, all right, and its replication cycle. And so we've got our chromosome here, and what we want to see is that there is one origin of replication, all right, on this chromosome, and it's right here, all right. It will be bidirectional, all right, and so it will go this direction, and it will also go this direction around this circle, okay. What that does is it speeds it up, and if you what you probably learned in terms of eukaryotes with these large chromosomes, that there are multiple origins of replication to get a chromosome completed. Well, in this case, we really only have one, and we notice that the terminus is on the opposite side, so each one makes it halfway around. All right, so pretty basic, pretty easy. We're going to create what is called the replisome. All right. The replisome is just the DNA polymerase and all the associated proteins associated with the replication complex. And so, you know, that big mass of proteins that's going to kind of slide along the template strands of our DNA, right? It's going to use both of them as a template, but it's going to basically cruise along there and synthesize our DNA. All right. The origins of replication will end up separating here. All right, and we can already see here is one, and here is our other. All right, and so our two origins of replication are going to separate. As the replication continues, right, our cell is still elongating and growing. So there's an awful lot going on here, right? This thing is growing like so. You're replicating the DNA. Once you get those two chromosomes separated, then that septation can occur. All right, so we're going to make that. We have our two separate cells, one over here, one over here. All right, and basically we can redo this process over and over and over and over and over and over. All right, and all told, it should really only take about 20 minutes. All right, so if the growth conditions are really good, you got 37 degrees, lots of nutrients, this is going to take about 20 minutes. Now, I say that, but what I want you to think about is I believe it takes about 45 minutes to replicate the chromosome. And so interestingly enough is that once these origins of replication are separated, right here, you can actually start replicating that chromosome a second and a third and a fourth time. And so when you have an active culture of E. coli going, you actually, when you separate these two chromosomes, right? So as in step 4 here, right? We've separated these two chromosomes. Well, in reality, this chromosome and this chromosome have already partially been replicated a second time, right? And that's how it kind of allows it to grow as quickly as it can. So if it knows the conditions, well, it doesn't really know anything, but if the conditions for growth are really good, that is what will be occurring inside of our cell. Now, this begs the question then, well, how is it that this cell, right, recognizes that it has gotten large enough to replicate the chromosome, that it's gotten large enough to actually separate the two cells, right? What's going to trigger cytokinesis? So we need to ignore this. This MREB protein, this is associated with cell shape, all right, and we'll get to that later. But what I want you to look at is this MinCD protein, all right, and this FTZ ring. The FTZ ring is the best studied of all the cytokinesis proteins, and when it forms that ring allows for the cytokinesis to occur. Now, MinCD oscillates back and forth, so it's going to come this way, right, and then it's going to work its way back this way, and it's going to go back this way, and it's going to oscillate back and forth across this cell, all right, and as it does that, it prevents this FTZ ring from forming in the center, all right, and if the cell becomes too long, what happens is that basically the MinCD doesn't pass through the center, and it's not going to prevent the FTZ ring from forming. All right, and so they've actually captured this in the form of these pictures using a fluorescent green protein that attaches to the MinCD. All right, excuse me. And so what happens is you can see it. All right, let me pick a color that'll show up here. So what you can see here is if you look at 0 seconds, all right, you can see this concentration of this MinCD on the poles. Okay, and what we want to look at in particular is this cell, all right, that it's here, and here, right? This is the same cell, and what you can notice is here's our MinCD on the right. It's oscillated back to the left. It's oscillated back to the right. All right, and you can actually see this occurring somewhat with the one below it as well. All right, and so it's this oscillation back and forth, all right, that is going to prevent the formation of that Z ring. Okay. Now, if we look at what we call the divisome, all right, and what the divisome does, oops, let's pick a better color again, one you can actually see, okay, so the divisome is the structure involved in actually, creating that septum and separating the two, all right, and so this is E. coli, and so this is our periplasm, right here. Here is our peptidoglycan layer, all right. This, sort of maroonish layer here. This is our membrane, okay. And then out here is our outer membrane. So we want to keep all of those structures kind of relatively clear in our minds. So what you want to notice then is that a large part of our divisome apparatus, right, is on the inside. Right, it's on the inside. It's on the cytoplasmic side of our plasma membrane. There are a number of proteins in the periplasm, right? These here that are anchored, okay, and they're going to have functions that will be more important in terms of peptidoglycan synthesis for the most part. Okay, so let's look at some of these structures then first. And so we want to look at is FTSA, all right, which is this one here. And we want to look at ZIP-A, which I believe is this one here. Now, what they do is they are going to anchor, all right, the Z ring to the plasma membrane. So they're going to basically anchor it right here to this plasma membrane and keep it from floating further into the interior of our cell. Okay. So we have those, ZIP-A and FTSA, and their role is to anchor. The FTSZ, right, that's these sort of, I don't know what you want to call this shape, sort of a crude airplane or, spaceship looking shape. But anyway, that's going to form this ring on the inside of our membrane, all right, and that's going to be sort of the controlling structure that forms, that allows for the divisome then to take on its final form, all right. The FTSK protein, right, which is right here, okay, this one is believed to be important in segregation of the chromosome and separation of the chromosome dimers, right? So when we have those two chromosomes being replicated, that we use FTSK to help separate those two, all right? Now, FTS, Q, L, and B, okay, these, they may be providing a scaffolding to allow for peptidoglycan synthesis, all right, so they might just be sort of a important structural, all right, and then we have FTSL and FTSW, all right, FTSL, this is our transpeptidase. And it is our penicillin binding protein, right? So this is the thing that penicillin will bind to and allow it to prevent it from making the wall, okay? Now, it is also my understanding that this FTSL, all right, in particular, can be very important in creating the gaps and the breaks in the wall in this peptidoglycan. Remember, we have to fracture it or cut it and insert new bits of peptidoglycan in there, and we certainly have to do that as the cell grows and enlarges, and we have to do this to to synthesize the peptidoglycan that's going to form that septum. Okay, the FTSN protein, all right, is right here, and it is believed that this one will trigger the constriction of the septum. All right, so those are the functions of each of these proteins that are going to assemble as part of this divisome process. Now you remember that the Z ring cannot form if the min-CD proteins are oscillating back and forth fast enough and spending enough time in the middle to prevent the Z ring from forming. Okay. All right. So there was one other thing I wanted to tell you, and what was it? I guess it was the min-CD. So anyway, that's the basics of the creation of the divisome structure to allow for cytokinesis to occur. Your text provides us with this nice table that summarizes the functions of each of these structures. Okay, and so what you want to be able to do then is you want to be able to basically create this structure, more or less, and you want to be able to discuss the functions of the particular proteins related to peptidoglycan synthesis. All right, this area, right? These proteins here are of a lot of interest to medical science, in that if you could design a drug that prevented the Z ring from forming, for example, so maybe it just inhibits ZIP-A, right? Will that actually then prevent cell growth? And in some cases it does, although they have found bacteria that don't require some of these proteins for their cytokinesis, but you have to wonder, would it be effective in preventing cell growth and therefore have a new class of antibiotics? So the one thing I guess that I was thinking of previously that I needed to tell you is that FTS stands for filamentous temperature sensitive. And what that refers to is that if you create these proteins so that they're sensitive to temperature and you denature them, the bacteria will tend to be filamentous or grow into, rather than say it's a rod shape. Well, then what you'll end up with is a really long cell that's more like a filament, and that's because, and it may even have multiple nuclei, or not nuclei, I should say chromosomes in it, all right? And normally you would divide this thing here and here and here and here, and it cannot do so because of the inability to carry out cytokinesis. And so this could be a powerful tool then in the fight against infectious disease. Now, if we want to look at peptidoglycan synthesis, all right, we want to really focus in on this ability to create gaps. All right, so here is my peptidoglycan down here. All right, here is my plasma membrane. All right, now and then here is my cytoplasm. Okay, so what we want to do is we want to think of, here we have a NAG. All right, so that is our N-acetyl or glucosamine, and here we have a NAM. All right, and to those, all right, are attached a uridine diphosphate. Okay, now what isn't shown in this diagram is that really there are amino acids attached to the NAM, right? And so we've got our, so we have our tetrapeptide, and I'm drawing this clearly too big, right? But you want to be thinking of, and there's actually 5 of them. One of them gets clipped off when we do the transpeptidase reaction, okay? So they've removed those just to make it easier to see. And so the NAM, all right, so let's just do the NAM first. We'll do this one, all right? It is going to be transferred to something called bactoprenol, and that will allow then we will lose the uridine diphosphate, all right? And so the uridine diphosphate is going to transfer the subunit, right, of our peptidoglycan, in this case the NAM, the same is true for the NAG, to the bactoprenol, all right? The bactoprenol then is going to carry this NAM-NAG unit, all right? So why they haven't shown this thing together, but anyway, they're going to carry those across, all right, and deliver it to the periplasm. Now, autolysins, all right, that's these A's here, all right, which are stimulated in the presence of penicillins, cephalosporins, all right, but even just during cell growth, all right, they're located at the divisome, is going to degrade the bonds in the existing peptidoglycan, all right. That will allow us to then, so if we break these bonds, okay, so we are then, boy, the way they drew this is, I don't like the way that this is straight, right, right, remember how these things are really kind of, you can't see the transpeptidase bond there, right? But so what we're really going to do is we're going to take something that looks like this, and this would be my NAM, and this would be my NAG, right? And to this I would have my peptide, right? And so I'm going to bring this whole thing here across the membrane, and I'm going to insert it. And so what I'm really going to do is I'm going to insert something that looks like this in sort of a half, all right? But I'm also going to bring this other side across, all right? Let's just say it's here for argument's sake. And then the transpeptidase, right, is going to make my final bond to this thing right in here, right? It's going to make that final linkage right in here, okay? So that's how this works, right? It's going to take some energy and some unique carrier molecules to get these large NAM-NAG complexes out and across the membrane and into the periplasm or outside the cell where you could actually synthesize this. All right? So that's what I have for basic cytokinesis.